Hello, my name is Naomi Golbin.
And I'm Ariella Golbin.
The focus of a research project is to identify and characterize the abscisic acid family of receptors in the fern Ceratopsis richardii.
Abscisic acid, or ABA, is an isoprenoid molecule that is produced in the chloroplast in all parts of the plant. In seeds, it promotes dormancy. Similarly, ABA restricts growth under extreme conditions such as high or low temperatures. To prevent excess loss of water during transpiration, ABA regulates the model opening. It also plays a role in reducing photosynthesis during stressful conditions where resources are limited and decreases cell division in times of stress. For a long time, scientists were not able to characterize ABA receptors because ABA is an unstable molecule with a half-life of about 24 minutes. The discovery of pyrobactin mimicking the activity of ABA led to the identification of the ABA receptors in 2009. Further characterization showed that there is a family of ABA receptors. These receptors are referred to as pyrobactin-resistant receptors, or PYR, pyrobactin-like receptors, or PYL, or regulatory component of ABA receptors, or RCAR. All of these belong to a large family. In the model sea plant Arabidopsis thaliana, this family consists of 13 members, collectively known as PYR, PYL, and RCAR.
ABA and pyrobactin are known to bind to receptors. The functions of individual ABA receptors are not all well characterized in various plants. Some of these receptors are known to act synergistically, while others work antagonistically. Generally, binding of ABA or pyrobactin to an ABA receptor located in the cytoplasm or nucleus initiates a signal transduction pathway that involves regulation of transcription factors and other factors such as the potassium pump.
A PYL molecule has highly conserved motifs, including the gate and latch domains. Before ABA binds to the receptor, the latch binds and the gate is opened. Once the ABA molecule binds to the receptor, the gate closes and allows for the interaction of other signaling molecules, such as phosphatases.
Drought and salinity affect pollen and ovule development dramatically. The pollen and ovule are drastically affected by drought stress, which leads to ovule abortion, pollen sterility, pollen tube deformation, and reduced yield. The pollen and ovule are very hard to study because they are hidden within the flower of the plant and are not free living. In order to study the gametophyte stage, we chose to use the gametophytes of the seedless plant of the fern Ceratopsis richardii. Gametophytes of this fern are free-living and two-dimensional, and therefore easy to study. We focus on the gametophyte stage to characterize the ABA receptors.
For our research, we wanted to ask how pyrobactin was perceived in the fern Ceratopsis richardii. We hypothesized that pyrobactin will inhibit the development and growth of the gametophytes in a dose-dependent manner, and that the perception of pyrobactin will occur through conserved ABA receptors.
The first question we addressed was how does pyrobactin affect the development of the gametophytes? We treated with pyrobactin only 20 micromolar data was significant in regards to the germination data. However, looking at cell number and area, the 10 micromolar as well as the 20 micromolar were significant. The cell number in regards to the 10 micromolar decreased by 20 or 30 cells, and the 20 micromolar decreased by 40 to 50 cells compared to the control. This suggests that a higher dosage is needed in order for there to be an effect on germination, while a lower dosage is able to affect cell division.
The next question we wanted to answer was how many ABA receptors are in the genome of Ceratopteris. In Arabidopsis, there are 13 ABA receptors. When we screened the genome with basic local alignment search tool, we found 7 putative ABA receptors. Several PYL receptors in Ceratopteris richardii have a homology of over 50% with PYL receptors in Arabidopsis thaliana.
The next question we wanted to study was where these ABA receptors are localized on the chromosome and how the gene is structurally organized. We found 2 ABA receptors on chromosome 32 with variations in intron and exon lengths. One of them contained a putative retrotransposon sequence in the intron. Similarly, an ABA receptor was also found on chromosome 12. This ABA receptor had a similar organization to those located on chromosome 32, with more retrotransposon sequences found in intron. 3 other putative ABA receptors are localized on chromosome 7, chromosome 18, and chromosome 30. These ABA receptors had no introns. One of these ABA receptors, located on chromosome 5, showed significant truncation in the coding sequence.
Next, we wanted to determine if all of these genes would code for full-length ABA receptors. To determine this, we found the putative open reading frames for all of the sequences, aligned the sequences, and mapped the various functional domains that are characteristic of ABA receptors using the known motifs found in Arabidopsis thaliana. A typical ABA receptor has a gate and a latch that allows for the binding of the PP2C domain once the ABA binds to the hydrophobic pocket and closes the gate. These are present with the exception of the truncated receptor referred to as 7100.
Phylogenetic analysis of the ABA receptors showed that some ABA receptors evolved before the separation of seeds from seedless plants, as they are orthologs found in seed plants. Several of the ABA receptors also evolved after the divergence of seed plants, as seen here. With regards to the truncated ABA receptor, it is most similar to the PYR and PYL1 in Arabidopsis thaliana.
The next question we wanted to address was if ABA receptors were differentially expressed and what conditions would regulate their expression. To answer this question, we examined the promoter sequences to look for possible regulatory DNA conserved motifs. We found the truncated ABA receptor 7100 contained a CpG island, which allows for gene regulation by DNA methylation. In addition, this ABA receptor has several conserved transcription factor binding sites, such as ABFs and ABIs, which respond to ABA.
To investigate if the truncated ABA receptor is regulated by DNA methylation, we treated the spores with an inhibitor of methylation called zebularine. After treating the metaphytes with zebularine and isolating RNA, we performed an RT-PCR. We found an increase in expression when treated with salt and zebularine.
Future studies would involve characterizing the expression of all of the ABA receptors during various stress responses, also to determine the role of each ABA receptor by performing a knockdown of each gene. Additionally, to assess if the truncated ABA receptor is a functional receptor or a pseudogene, and can 7100 bind to ABA and interact with other proteins. In conclusion, similarly to seed plants, the seedless fern can perceive pyrobactin and contains a multi-gene family of ABA receptors. The function of each receptor remains to be determined. One of these receptors codes for a truncated protein and possesses many regulatory control mechanisms in its promoter region. It also is repressed by DNA methylation.
Rationalize their references. Thank you for listening to our presentation.